जैसा कि हम पहले रिएजेंट्स और सप्लाइज के हवाले से बात कर चुके हैं, इक्विपमेंट के हवाले से बात कर चुके हैं कि हमें डीएनए प्योरिफिकेशन के लिए क्या क्या चीज़ें अरेंज करना है. वंस ऑल दोज रिएजेंट्स आर अरेंज्ड, देन वी कैन proceed with the procedure. As we are going to use phenol is a toxic chemical, so therefore we have to use gloves, and the phenol treatment should be done in fume hood. The step-by-step treatment of the DNA is like we have to take the DNA sample and an equal volume of phenol will be added in it. If we take 50 microliter of DNA sample, this is a very little volume, so we have to add 50 microliter of TE buffer at pH 8. Or if we have starting material 100 microliter, we can simply add 100 microliter of phenol in the DNA solution. At this stage, we have to mix the phenol and the DNA sample, but we cannot vertex. Vertexing can damage and break the DNA into pieces. So by inverting the tube gently, we can mix the two different contents, and once the mixing is done until an emulsion is formed, until that time we have to keep inverting. After that, centrifugation at 14,000 rpm for 20 seconds at room temperature will be done. As a result of centrifugation, we will see two phases. The upper one is the aqueous phase, the lower one is the phenol, and at the junction there will be present the proteins. Very carefully transfer the upper aqueous phase to a new tube, the microcentrifuge tube. Now repeat this extraction process so as we make it sure that maximum DNA has been extracted from the sample. For this purpose, add 100 microliter of TE buffer, mix it gently with the aqueous phase, mix it gently by inversion, centrifuge as in step number 4, that is 14,000 rpm for 20 seconds, and remove the aqueous phase and shift it to the previous aqueous phase. Now we have 200 microliter of the extracted DNA. This is going to be treated with the second step, the chloroform and phenol. 200 microliter of the DNA is going to be supplemented with 100 microliter of phenol and 100 microliter of chloroform. That is the equal volume. Again, we have to mix them, but there will be no vertex using. The mixing will be done by simple inverting the tubes, and after mixing we have to follow the centrifugation at step number 4, that is 14,000 rpm 20 seconds. Once the centrifugation is done, two layers will be formed. Again, remove the aqueous layer and shift it to a new tube. At this stage, now we have to use only chloroform. 200 microliter chloroform will be added into the extract, and it will be mixed by inverting the tube. Again, after mixing, centrifugation at 14,000 rpm for 20 seconds, and two phases will be formed. The upper aqueous phase contains at this step highly purified DNA. Remove the upper aqueous phase and store it at 4 degrees centigrade for further usage, or on ice if it is immediately required for further usage.